Hello, my name is Jeff Birch, and I'm a supervisor in pest detection for LA County Agricultural Commissioner. I will be going over what pest detection is, what we do, and why we do it. The outline of the presentation starts with how pests are handled in different divisions. We will then talk about traps, then specific insects, and go into trapping procedures before we finish up. So this is how handling pests is broken down into different divisions, whether it be at the county, state, or national level. Pest exclusion and plant quarantine is the first line of defense. They do their best to block the pests from getting in. They use methods like phytosanitary certificates, inspections of nurseries and wholesalers, and dog teams. But sometimes a pest will slip through, especially in Los Angeles with so many potential avenues of introduction. We have several major airports and two international seaports on top of all the mail parcels that get routed here. If a pest does find its way into LA County, it is pest detection's job to find that pest, to locate any introductions or infestations. Pest detection is typically coordinated by the County Agricultural Commissioner through a contract with the state. The goal is early detection before the pest becomes established. Once pest detection locates a pest, then pest eradication comes in to get rid of the pest. Pest eradication is initiated by the California Department of Food and Agriculture, or CDFA, which involves placing more traps, removing hosts, and potentially pesticide treatments. The primary method used to detect insect pests is by traps. Traps can be used to monitor and eradicate pests. The basic idea of a trap is to have a lure to attract the insect and to have a way for the insect to enter the trap but not be able to leave. The lure can be several things like food, visual clues, or a chemical like a pheromone. Traps can hold insects using sticky surfaces, liquid, or mechanical obstruction. These traps listed are the different types of traps we use in LA County, with the length of time they are deployed at each site. There is the McPhail or wet trap, the Jackson or dry trap, Japanese beetle trap, and gypsy moth trap. The McPhail trap is one of our fruit fly traps. It is a general fruit fly trap with a food-based lure. We use it for Mexican fruit fly because there has been no effective chemical lure developed for Mexican fruit fly. The fly is attracted by the smell and goes into the opening in the bottom of the trap. As the fly tries to escape out the top by following the sunlight shining through the glass, it will eventually get trapped in the liquid. We use Jackson traps to detect Mediterranean fruit fly, Oriental fruit fly, and melon fruit fly. Each of these fruit flies has a unique pheromone lure that is placed into the trap. So Jackson traps are specialized for the species, in contrast to the McPhail trap that attracts all fruit flies based on a feeding response. The trap pictured is set up as a Mediterranean fruit fly trap. You can tell this by the lure in the middle of the trap, which is the pink puck and all the Mediterranean fruit flies below the lure. Here is the anatomy of the Jackson trap. Again, this is for Mediterranean fruit fly, and the lure is a solid puck with no pesticide inside the basket. Then you have the hanger, which hooks onto a branch in a tree, the overall body of the trap that contains all the components and where all the trap information is written on. Then you have the insert on the bottom that is coated with glue to catch the insects. The insert is removable so that an inspector can easily examine and replace it. This Jackson trap is used for Oriental and melon fruit fly. The only difference is the lure. Since the lure is liquid, we use a cotton wick to absorb it. The lure is a pheromone, specific to the species, and it's mixed with a small amount of the pesticide dibrom. Dibrom is used to stun the insects when they enter the trap so they fall into the sticky insert. Next is the Japanese beetle trap, which you'd be surprised to find out is used for Japanese beetles. We place it out in the summer months, and it is hung from a metal pole that is stuck in the ground. The main hosts that we try to place our traps near are roses and turf. What's cool about this trap is that it employs 3 different types of lures to attract the beetle. It has a feeding lure of phenethylpropionate, eugenol, and geraniol, which smell like roses. It has a pheromone to attract males called japanilure, and the green color of the trap is a visual attractant. The beetle will fly towards the lure, hit one of the fins in the top, and get funneled into the can at the bottom. An inspector removes the can to check for targets. If a live beetle is found, it is submitted in an alcohol vial to kill it. If a dead beetle is found, it is submitted in a dry vial. CDFA will place target specimens called plants in our traps to make sure we are doing our job. So if the beetle is dead, it could be a possible plant and can be tested for the presence of alcohol to verify it as a plant. That is why dead beetles are submitted in dry vials. The gypsy moth trap for gypsy moth is hung on trees with wire. Broadleaf trees are preferred, but they can be hung on conifers as well. It is also placed in the summer at the same sites as Japanese beetle traps. The pheromone disbarlure is soaked on a string and attached to the inside of the trap. The other two interior sides are coated with glue. Now we will talk a little bit about each of the target pests and see what they look like. The Mediterranean fruit fly is one of the world's most destructive fruit pests. It feeds on over 300 different fruits, vegetables, nuts, and flowers. It is successfully controlled in Los Angeles County by a male sterile release program. The program is a biological control method that rears large numbers of sterile males, releases them into the environment, they mate with females and produce no offspring, thereby declining the population. All the fruit fly traps are serviced year-round. Medfly can be identified by the golden band across its thorax and the gold-brown wing patterns. Oriental fruit fly's lure is the pheromone methyl-eugenol, and it is mixed with 1% of dibrom. It is a green color. The main hosts are tropical fruits, stone fruits, and citrus. Oriental fruit fly's identifying characteristics are the black T on the abdomen, the darkened edges of the wings, and the two yellow stripes on the thorax. This picture shows how fruit flies are agricultural pests. These female oriental fruit flies have their ovipositors inserted into the fruit, depositing eggs. Once the egg hatches, the larvae, or maggot, feeds on the fruit until it matures and falls to the ground to pupate in the soil. This is why it is important to control these fruit flies, because they can cause this damage to the hundreds of hosts they target. Melon fruit fly's lure is Q-lure mixed with 5% dibrom, a slightly higher concentration than oriental fruit fly. The color of the lure is brown. As the scientific name implies, the favored host is fruit from the family Cucurbitaceae, which includes squash, watermelon, and cucumber. The melon fly also has the black T on the abdomen. The wings have two dark spots, and the thorax has three yellow stripes. The Mexican fruit fly is trapped with the McPhail trap that uses a mixture of yeast, borax, and water. It is the largest among the target fruit flies and has an overall yellow-orange color. It has three yellow stripes on the thorax. And the yellow-brown wing banding form an S and a V. In Los Angeles County, we don't specifically trap for Caribbean fruit fly, but we do find it in our traps, and it requires eradication when found. Caribbean fruit fly has similar wing banding to the Mexican, except it is darker in color. There is also a characteristic black spot on the thorax. We trap Japanese beetle during the summer. We try to catch the beetles as it emerges from its pupae in the soil. They will eat hundreds of different agricultural and ornamental plants. The larvae are particularly fond of turf, and the adults like roses. Japanese beetles have metallic green bodies with copper-colored wings and white hair tufts on the abdomen. They are a devastating pest on the East Coast, and every year happen to make their way onto airplanes and hitch a ride to LA. CDFA even has a program to inspect planes for these potential stowaways. This insect is actually a fig beetle. Fig beetles are common in California and are frequently mistaken as Japanese beetles. Before I started working here, I called these guys Japanese beetles too. Looking at the two side by side, you can see the drastic difference in size and coloring between the fig beetle and Japanese beetle. Gypsy moth is a major pest of trees. It is the most destructive pest of hardwood forest and shade trees in the United States. The damage is done to the leaves by the larvae. The adults actually don't feed. You can see the major differences between male and female gypsy moths. They both can be identified by the black crescent mark lateral to a black dot on the wings. But it is more distinct in the female. The male is smaller and has large feathery antennae to pick up the scent of females. The eggs are laid in masses of 100 to 1,000 and are covered with hair, forming a soft tan patch about the size of a quarter. In infested areas, egg masses are found on anything kept outdoors, from furniture to lawnmowers. If you move from an infested area and bring these items with you, you can potentially introduce this pest into a new area. Gypsy moths have cool-looking caterpillars with a distinctive color pattern. It has 5 pairs of blue dots and 6 pairs of red dots along their backs. This is a great picture of the antenna in action on the male. My division does not deal directly with Asian citrus psyllids, but our department and the state definitely does. It is a very dangerous pest of mainly citrus species. ACP is so important because it vectors the pathogen that causes Huang Long Bing disease, HLB for short, and also known as citrus greening. This plant disease is incurable and causes trees to die within 10 years. HLB is the most serious threat to citrus trees worldwide. The Asian citrus psyllid is small, about 3 to 4 millimeters long, with a brown mottled body. The head is light brown and the eyes are red. The insect is covered with a whitish, waxy secretion, making it appear dusty. It feeds at a unique 45-degree angle, as can be seen in this picture. The nymphs can be identified by the curly white waxy tubules it produces. The nymphs excrete a large amount of honeydew, and the tubules help clear the waste away from its body. Asian citrus psyllid was first detected in the United States in Palm Beach County, Florida, in June 1998. It was first in California in August 2008 in San Diego County. ACP has expanded into the Central Valley, Central Coast, and has been found as far north as Sacramento. Much of the spread is due to movement of infested nursery plants. HLB was first found in California in 2012, but is not spread outside of Southern California. The damage caused by ACP alone is leaf twist, killing new shoots and helping growth of sooty mold. This picture gives you a better idea of the actual size of Asian citrus psyllids. This is the leaf twist caused by ACP. The early symptom of Huanglong Bing is leaf yellowing in just one section of a tree. The yellowing is asymmetrical and mottled. Over time, the fruit becomes smaller, lopsided, may stay partially green or drop prematurely, and the juice becomes bitter. Eventually, the foliage becomes sparser from limb dieback, the tree stops bearing fruit and dies. Survey methods for ACP include visual inspections of citrus trees, sweep netting for insects, and the placement of yellow panel sticky traps in host trees like in the picture. In response to ACP and HLB in California, CDFA began an extensive monitoring program to track the distribution. They check thousands of yellow sticky traps and frequently test ACP and leaves for the presence of HLB. ACP quarantine zones have been established throughout the state that restrict movement of citrus trees and fruit in order to prevent psyllids from being moved to new, uninfested areas of California. Additionally, more restrictive HLB quarantine zones have been established to help keep HLB from spreading. Citrus trees and close relatives that could be hosts of the psyllid can't be taken out of quarantine areas. Fruit can be moved, but only if it is washed and free of stems and leaves that could harbor the psyllids. Species of tiny parasitoid wasps have been brought to California to reduce ACP populations. However effective, they have not eradicated ACP nor stopped the spread of HLB. In new areas where ACP has been discovered, CDFA conducts residential insecticide treatments to all nearby host plants. The treatment lasts for several months. Now we will get into trapping procedures, how we select hosts, place traps in those hosts, and when we relocate the traps. I will also describe piggybacking and delimitation. Selecting a trapping site depends on two main criteria: a suitable host and maintaining uniform trap distribution. First consideration should be a site with a suitable host. Each pest has a host preference list determined by CDFA based on research. Primary hosts are exceptional, secondary hosts are good, and so on. You always want to try to place a trap in a primary host, but just being the right host species is not enough. The host has to be able to accommodate the traps. A small sapling may not provide enough shade for traps, or certain hosts may have branches that cannot support the weight of the traps. If you have a suitable host that cannot accommodate the traps, many times you can proxy trap by placing the trap in a non-host tree that is within 20 feet of the suitable host. The stronger the bait, the farther the trap can be placed from the host. Trimed lure for medfly is one of the weaker baits we use for fruit flies. Therefore, when we place traps with different baits on the same tree, we always choose the best host for medfly since its bait is the weakest. We want to use the superior host to draw in more medflies. Fruit fly traps should be placed in the warmest part of the tree, but not in direct sunlight. It is best to place the trap in the upper 1/2 to 1/3 of the tree canopy and 1/2 to 2/3 the distance from the trunk to the outer edge of the foliage. Again, that's the upper 1/2 to 1/3 of the tree canopy and 1/2 to 2/3 the distance from the trunk to the outer edge of the foliage. It is also best to have fruit nearby. There should be a foliage-free space of about a foot around the trap so that the entrance is not blocked and the fly does not have a resting place close enough to the trap to be able to check it out without entering the trap. We want the fly to enter the trap so that it is knocked out by the pesticide and falls into the sticky insert. The same placement rules apply to McPhail traps. Put in the warmest part of the tree with filtered sunlight, near fruit, and no obstructions around the trap. Japanese beetle traps should be placed in the open, away from trees, houses, and fences. The trap can be placed at differing heights. If using only turf, the trap is placed 1 to 2 feet from the ground. If using a low-growing host like roses, the trap should be placed at the height of the host and 10 to 25 feet away from the host so the trap doesn't directly compete with the host. Gypsy moth traps should be placed 4 to 5 feet from the ground on single trunk trees with a diameter of 1 to 2 feet. When multiple traps with male attractants are placed in the same host, a distance of at least 10 feet should be maintained. This is to prevent competition between the lures. We place traps in warmest parts of trees because fruit flies don't like the cold, but not in full sunlight because it will degrade the pesticide. This diagram shows a typical fruit fly grid. It has an outer dimension of 1 square mile that is evenly split into 5 subgrids, or quints. Since fruit fly traps have a limited drawing range, they have to be relocated to effectively cover the quint. Traps need to be relocated every 6 weeks, which is the average life cycle of a fruit fly. Traps should be relocated at least 500 feet from the previous site and any other fruit fly trap site. Piggybacking traps just means that more than one type of trap is used on the same property. In LA County, we have over 18,000 traps deployed. It would take an incredible amount of manpower to service all these traps on different sites. So in order to economically maintain all the traps, we piggyback them. This diagram shows how we typically piggyback. Each quint has a McPhail trap and a piggyback site with the Jackson traps for Mediterranean, Oriental, and melon fruit fly. There is also 2 separate sites per square mile that has a piggyback gypsy moth and Japanese beetle trap. If a target insect is found, it is extremely important to determine if an infestation exists and how far it has spread. This is the purpose of delimitation surveys. It is part of the pest eradication step. Each target fly has its own delimitation requirements. The diagram above shows the medfly delimitation, which is one of the more complex. Each square is a square mile grid. The center grid includes the initial find site. So you can see in this grid, 100 champ traps and 25 multi-lure traps are deployed in this grid. Champ traps are yellow sticky panel traps similar to the ACP traps, and multi-lure traps are basically plastic McPhail traps. Then surrounding the core grid, 50 champ traps are placed in each square mile for a total of 400 traps. The traps in these first 9 grids are placed within 24 hours of confirming a wild fly. The 2-mile buffer grids each have 25 Jacksons, for a total of 400 Jacksons, and the next buffers decrease by 5 Jackson traps per square mile. I will show you the delimitation surveys for each of the major pests we trap for in LA County. Oriental fruit fly has 25 Jackson and 25 McPhail in the core grid, and then 5 Jackson in each grid surrounding the core up to a 4-mile buffer. Melon fruit fly has 50 Jackson and 25 McPhail traps in the core and 25 Jackson in the surrounding grids. Then the number of Jackson decreases by 5 for each buffer up to 4 miles. Mexican fruit fly just uses McPhail traps, 80 in the core, 40 in the first buffer, then 5 in the remaining buffers. Japanese beetle has 50 Japanese beetle traps in the core, then 25, then 5, and each surrounding buffer, only out to 3 miles. Gypsy moth delimitation is slightly different. Instead of a square mile grid surrounding a find site, the find site is an epicenter surrounded by 4 square miles of 25 traps per square mile. So the small black filled square is a single point where a wild gypsy moth was found. And that's everything. If there are any additional questions, please feel free to email me. Best of luck on the exam.